So today I will be speaking about nuclear myosin 1 and its role in transcriptional reprogramming adipogenesis. So not about microbiome, but I'm sure in future microbiome will be important part of our project. So with that, I start. Our lab is interested in genome organization, and of course, it's a very complex subject. But to make it simple, we are interested in role of nuclear cytoskeletal proteins in regulation of co-expression of a subset of genes, so-called gene programs. And these gene programs, we are mainly interested in those programs that are involved in development and differentiation and metabolic reprogramming. So today I focus on NM1 or nuclear myosin 1 that our team is working with for many years. So we have a lot of publication about that. And today I talk about a small part of our finding about NM1. So NM1 is a splice variant of, or isoform B of canonical myosin 1C. And it's shown that it's interacting with polymerase 1, 2, and 3. And the way that it works is that it's binding to transcription start site in a GSK3 beta manner, dependent manner, and followed this, there is assembly of chromatin remodeling complex B-Witch, and with the onset of this chromatin remodeling complex, the histone modifier like CET1 or PCAF can initiate the transcription in presence of NM1. So this is like a global role for NM1 in gene regulation. And recently we show that nuclear myosin 1 or NM1 is regulating cellular metabolism. So we have this cell model that is lacking NM1. They are mouse embryonic fibroblasts, and we did bulk RNA sequencing on those cells, and we noticed that a lot of genes related to oxidative phosphorylation, which is a mitochondrial respiration, is downregulated in those cells that are lacking NM1. And this is very important because if you imagine we have less mitochondrial respiration, then it means there is another type of respiration, which is cytoplasmic, and it is aerobic glycolysis. And this is actually indicating that there is some abnormalities in mitochondria. And this is normally happening in highly proliferative cells like cancerous cells. But it's not only in cancer cells, it's also important for cells that are located in the part of our body that there is not enough access to oxygen, for example, stem cells. And it's important for a stem cell fate decision. So we showed that if you stain the mitochondria, and one of the important protein for mitochondria, TFAM, you can observe that in wild-type cells, the mitochondria is very healthy, elongated, and spaghetti shape. But when you remove NM1, the mitochondria turns into abnormal phenotype, and it has this circular shape. But when you put back the NM1, again, the cells start to rescue their phenotype, and they have this spaghetti shape mitochondria. And we showed that NM1 is actually regulating mitochondrial transcription factors in an mTOR signaling dependent manner. And if you remove NM1, then of course we have these abnormalities in mitochondria, we have this imbalance increase in aerobic glycolysis or Warburg effect, and we shift the fate of cells toward cancerous cells. And in fact, if you take those cells, MEF cells that are lacking NM1, and inject them to nude mice, they start to develop cancerous tumors. And here you can see this study that we did. So knowing the important role of NM1 in regulating transcription factors and also its important role in chromatin remodeling, we wanted to look at data that we had from ATAC-seq and bulk RNA-seq data. So we integrated these two data, and we generated this plot that is actually looking at the chromatin accessibility where the chromatin is open and when there is gene expression happening actively. So here in this quadrant, you can see that we have higher accessibility to chromatin, and there is higher expression of genes in presence of NM1. And the opposite quadrant, you can see that there is less accessibility to chromatin and, of course, less expression of genes in absence of NM1. So you can understand how NM1 is important for opening of chromatin and starting the expression of specific genes. So further, we applied some machine learning approaches to data that we had from ATAC-seq, but this time we changed bulk RNA-seq to single-cell RNA data. And we computed a predictive model that is shown here as a network, and as the hubs are actually transcription factors. And the network around each hub is the genes that are regulated by that specific transcription factor. So we look at this list of transcription factors that actively involve in gene expression in presence of NM1, and we choose top high impact transcription factor, and we look at their expression in bulk RNA-seq data in absence of NM1. And we noticed that among top 10, 20 transcription factors, all of them were dysregulated in absence of NM1. And it turned out that most of these, almost all of them, are involved in adipogenesis or adipocyte differentiation. So now we know that NM1 is involved in gene regulation, and of course, it's regulating some transcription factors related to adipogenesis. We wanted to know how adipogenesis is changed or altered in absence of NM1 in our animal model that is lacking NM1. So we isolated mesenchymal stem cells, which are basically multipotent stem cells, and they can differentiate to some other cell type. And we took those mesenchymal stem cells from compact bone. We differentiated them to adipocytes, and we noticed that when they are mesenchymal stem cells on day 0, they don't look very different between knockout and wild type. But after 10 days, which is earliest stage of differentiation, we noticed that the cells that committed to adipocytes are significantly bigger compared to wild type. Although the number of cells that committed to adipocytes, they are less. So we had less cells, but the cells were bigger. And by day 20, that the cells were mature, still the same trend. The cells were bigger, the adipocytes were bigger, but there were less number of adipocytes. Then we look at some key regulators of adipogenesis. I'm sure all of you know about them, PPAR gamma and CEP alpha and beta. So here we have another animal model that is carrying one copy of NM1, and it's heterozygous. So we noticed that PPAR gamma and CEP alpha, both of them were downregulated in the absence of NM1 in heterozygous and knockout sample, but it was a slightly increased, CEP beta was slightly increased in a knockout sample. So this was actually indicating that we have impaired or stalled adipocyte differentiation process, because this PPAR gamma and CEP alpha are very important for early stage of adipogenesis. And then we look at 3 more key regulators that are FAP-P4, LPL, and adiponectin. And all of these 3 key regulators that are actually important for mature adipocytes were also downregulated in a dosage-dependent manner in the absence of NM1. And this is showing that there is a failure in terminal adipocyte differentiation and function. So now we know that NM1, in the absence of NM1, adipogenesis is impaired, or let's say altered. But what about the animal model? So we dissected our animal model, and we noticed that both in dorsal, so around shoulder, and also in abdominal region, there is more accumulation of fat tissue. And then we look at the weight of our mice, and we notice that the ones that are knockout, they gain more weight during their life compared to their wild type counterparts. So with this, we wanted to also check if there is actually more fat tissue inside the vital part of body, that is thorax and abdomen. So we performed CT scan, and we could confirm that there is a higher amount of fat tissue inside thorax, around heart, lungs, and so on, and also in the abdomen of those mice. Then If you remember, I said that these committed adipocytes from MSCs were bigger in knockout sample. What about the tissue? So we isolated the tissue and we look at the cell size and we noticed that there is the same trend. So the fat cells in the tissue were significantly bigger compared to wild-type tissue. And this is actually showing that there is a shift toward hypertrophy, which is an unhealthy way of cell growth. Then we performed some bulk RNA sequencing on tissue that we isolated, and we found that around 1,500 genes were differentially expressed between wild type and knockout, of course, significantly. And we look at the gene ontology, and we notice that interesting Go terms like PI3K, ACT signaling, inflammatory response, and angiogenesis are enriched, which are showing some tissue remodeling. Further, we look at the KEK pathways, and here again there are some interesting pathways like endocrine resistant, again PI3K, ACT signaling, and RAP1 signaling, which is actually showing a shift in cellular metabolism. And finally, a lot of genes that are shown that that they are involved directly in obesity, they were also dysregulated between knockout and wild type. And finally, this is very last slide. So if you remember the network that I showed you and the role of NM1 in regulating those top 20 transcription factors, and they were dysregulated in cells that are lacking NM1, and we noticed that they actually follow the same trend in tissue. So basically, what we found in MEF cells, it translates into tissue and animal model. And actually, we found that 3 of these transcription factors directly or indirectly can regulate mitochondrial biogenesis. And with this, I want to conclude and give you an overview of the project. So when there is NM1, we have this healthy mitochondria and healthy oxidative phosphorylation. We have healthy and, of course, a good number of adipocytes, and we have this slim, healthy mice. Then we have, when we don't have NM1, the chromatin is compact. We don't have enough transcription of mitochondrial transcriptional factors, and then we have this impaired adipogenesis, and finally the cells get bigger to compensate, and we have this obese mice. And with this, I would like to say thank you to Valentina and Raul, because they really helped me a lot with the mice work, and then, of course, my supervisor, Pier Giorgio, that is here, and, of course, everyone else that helped me in this project and supported me, and, of course, from you for your attention.